Good morning, everybody. Thank you very much to the organizers for this invitation. So actually, I come from University of British Columbia, Vancouver. We don't have many vineyards in Vancouver, actually none. But we do have a wine research center that works. We have 3 faculties working on grape and fermentation, particularly yeast genomics. And we work particularly with Okanagan Valley, that is our major viticultural area in British Columbia. However, I just relocated there a year and a half ago. So today I'm going to present you data that I've been producing here in California when I was a PhD student, and later on in Italy when I went back. And actually, I come from the same region than Alessandro Zanuta. So ironically, we met here miles away from home. So today I'm going to talk about the impact of water deficit on fruit composition and try to squeeze different studies considering both red and white grapes. We do know several things about water deficit, about the physiology and how the vine respond to deficit, less vigor, less transpiration and photosynthesis, particularly if the deficit is severe. Increases in ABA. ABA is the plant hormone that signals stress. And this is quite interesting because it's also the hormone that we think triggers ripening or modulates ripening in the grapes. There is unfortunately, a reduction of fruit growth. Well, unfortunately, because we lose yield, but in some cases, because we increase the ratio between the skin and the flesh, we have an increase of the metabolites accumulated in the skin, so that could be good for grapes. And but what is actually the focus of my research, of my lab, is water deficit might impact the metabolism of the fruit, and we focus on secondary metabolites. Some are the metabolites that we have always studied, phenolics. Now we're moving toward volatile compounds. So the objective of the lab is kind of understanding how deficit modulates the whole metabolism. We study also the expression of the genes that control this metabolite with the same technology that Dario Cantu has explained before, but today I'm not going to go into those details. So briefly, we do know that there are many metabolites that affect quality. Some are volatiles, like terpenes, non-isoprenoids, methoxypyrazines. Others are not, and are particularly phenolics. Here I mentioned the major ones for fruit quality. Most of them are accumulated in the skin, and in some cases, like catechins, or are also called flavantriols, and proanthocyanidins that are called tannins, are also in the seed. So, as I said before, we know that water deficit, and we can observe easily that water deficit reduces fruit growth, berry size, and we know that this might lead to an enhanced concentration of metabolites that are produced in the skin. And it was the first study I did, it was here actually, as I said, in California. I'm not explaining too much about the study, but it was Dunnigan Hills. The trial was hosted by R.H. Phillips, and we had this trial that was aimed to investigate if water deficit was actually affecting the production or the biosynthesis in the cells of phenolics or of anthocyanins. And so we studied 3 different irrigation treatments. Control were irrigated grapes. I'm not going too much in details. I have a slide for the question time if you are interested. Early deficit, stress before veraison, quite severe stress, and then late deficit, quite severe stress after veraison. And this is something that was kind of already known from previous studies at UC Davis. So water deficit stimulates the accumulation, the concentration of anthocyanins in the grapes. What if particularly early deficit that was kind of surprising for me. We apply deficit before anthocyanins are accumulated, but we see a boost of anthocyanin accumulation at veraison. The curve with the triangles is the early deficit one, and you see that kind of anthocyanin maximum is reached very quickly, much faster than in control irrigated vines and late deficit vines, so vines that have been stressed from varaison on. And this actually is connected to other things we see, and I'm not presenting on another variety, Merlot. We try to see the effect of early deficit on varaison time and the speed of varaison, how fast they progress from green berries to red berries. And we see, actually, we don't see much of an enhancement of varaison date, but we see a much faster progression of varaison and color development under early deficit condition. However, the goal of this study was to understand if there was a direct effect on the biosynthesis, or if it was just a matter of concentration of these compounds. So don't go through this graph. Maybe it's better to summarize it. Those are all genes that are controlling the synthesis of anthocyanins in the grapes. Not only anthocyanins, flavonoids, but some of them are specific for anthocyanins. And if you see, for instance, UFGT picks up at varaison and controls the anthocyanin production, is the last one here. Basically, we studied the gene expression to understand if there were the cells that were going under drought condition were producing producing more anthocyanins inside. And so I'm summarizing the graph here. What we observed was that under early deficit condition, this pathway was triggered early on, some days earlier, and went up faster than the other. And then during ripening, we kept observing higher level of expression of most of the genes of the pathway in the berries of vines that were stressed, both early and late deficit. So is here, while the control is the line down there. So there is a direct effect of water deficit on the production of these compounds in the cells. It's not just a concentration effect. This was the first thing and regarded like anthocyanins, and then I moved on with another study. This study is on Merlot. It's back in Italy, in Northeast, where I was working at the University of Udine before moving to Vancouver. And it meant to analyze more in general the metabolism of the fruit and how the metabolism can be affected by deficit. We have Merlot vines, 20 years old. We have like vines that we irrigate and other vines that we stop irrigating. They go through severe water deficit for about 40 days after anthesis, and we kept the stress quite low. We like to stress our vine a lot because we want to see major effects of the deficit. Then we'll need to do other trials to translate this into a more applied techniques though. So we did 2 years of study. I think you are familiar with stem water potential or leaf water potential is what we use. And here you can see that controls stay high in leaf water potential or stem water potential. Deficit vines go down, and it starts before varaison, but it gets really severe after varaison. And so this time, so what we did this time, it was not just to look at specific targets like anthocyanins or total tannins. We tried in collaboration with another lab in Italy, to do a profiling of hundreds of metabolites, secondary metabolites, volatiles, phenolics, stilbenes, resveratrol, these kind of compounds, and how the effect was modulating these metabolites and the concentration. So the first thing I'm going to show, though, is the data at harvest, and so those are Merlot vines, and we observe a decrease in yield, a decrease in berry size. It's 30% decrease in yield, so it's quite severe. We see a big decrease, and then also an increase in sugars. Sugars level are much lower than the one that you probably observe in California. And this is because it's northeastern part of Italy. It's a cooler area. We don't get very high Brix there. But now I'm going more into the impact of water deficit on the metabolism. And I know this is not a friendly user slide, but it's like how we like to represent the data when we have a lot of metabolites and a lot of data points. In this slide, you can see 2011, 2012, and then there are the day after anthesis where the samples were collected. So there is a number, I think is from about 30 days and then till harvest. And for both years, each line is a metabolite that you cannot read, but I summarize the category close by because I know that it's difficult. And then at each of those squares are actually like the fold change, increase or decrease of that metabolite under deficit condition. For instance, if you have like the a red square, it means like that there was the double amount of that metabolite under deficit condition. If you have a blue square, it means that it was half of the amount under deficit condition. And I'm basically summarizing. You see that a lot of metabolites have asterisks. That means that they were significantly affected by water deficit. The concentration was affected by water deficit. And this is what we observed in Merlot quite consistently between the two seasons. Anthocyanins, we knew that they increase. We have seen an increase in benzoic acid, in gallic acid, an increase in cinnamic acid concentration like aphtharic acid. So those are small phenolics that are accumulated in the grapes, probably the major one in white grapes, not the major one in red grapes. We did observe like a decrease in catechins or flavantriols, so the monomers that they build up the tannins. It's not here, but we did observe like a small increase in the total amount of tannins. 5 minutes? Okay. And good. And also still being resveratrol, you all know that they go down. And this is different from what we have seen in Cabernet Sauvignon. Not we, other groups have seen in Cabernet Sauvignon. So different varieties might respond differently. We also did like some analysis of volatile compounds. We see less effect of water deficit on volatiles. I go straight to the point, but we have seen some volatiles that are aldehydes, ketones, and alcohols, small chain alcohols or aldehydes that are increased in concentration under deficit conditions. And however, as far as I know, we don't exactly know. We know that those metabolites are in the fruit, are in the grapes, are also in other fruits like tomato, strawberry, but I don't think for those particular ones, we can associate a given aroma. However, when we produce the wines and we evaluate the wines, those are Merlot wines, and we found what was often found that if you have a stress vine, you get like more phenolics, or potentially a bigger body, and more fruity, jammy, and other aromatics. And these aromatics might be connected to the one that we have seen that are increased, but we cannot say that yet. Interestingly, we have also seen, of course, more color, but also change in hue toward blue hues instead of red, pink hues, and this is consistent with the higher production of some specific anthocyanins like malvidine and delphinidine and peonidine. So we find some consistency in what we see in the fruit and then in the wine, of course. So I'm moving fast into another project we did on white grapes. Now my lab is kind of moving toward white grapes. White grapes are important for Okanagan and for many other regions, and deficit is something that, despite the general wisdom that deficit is bad for white grapes, we don't have a lot of publications, so we want to see what happens in the fruit, in the metabolism of the fruit. So here is a similar experiment than before with a variety that's called Tokay Frulan or Sauvignonas or Sauvignon Vert in Chile. So it's quite popular in northeastern part of Italy. Similar project, big deficit before, well, not too much, but quite severe, very severe deficit before variation. We had to irrigate our deficit treatments, even though the area is not normally so dry, because it was going to far the deficit. Of course, we see a decrease in the size of the fruit, a decrease of yield, and we have seen again this year with this early deficit an increase also in the sugar levels in the fruit. And I'm going to quickly through the metabolism. It's a similar graph when you have a red square, higher concentration. If you have a blue square, lower concentration, and this is a fold change, so red means double amount, so it's quite big. On the phenolic side, we know white grapes do not produce anthocyanins. But we see a similar effect than what we have seen before, higher levels of cinnamic acid and benzoic acid, gallic acid, and caphtoric acid particularly. And we do not see any, we have some of the catechins went down, but normally no big effect on that. We have studied also the carotenoids. I didn't mention them. Carotenoids are secondary metabolites that protects the photosystem apparatus of the berry when it's green. And they are quite important for quality because the degradation leads to neuroisoprenoids, beta-damascenone, alpha-ionon. So we see that they go down under deficit, so we assume more beta-damascenone or beta-ionon. But we don't see that. Actually, the neuroisoprenoids were the same. But we have seen an interesting phenomenon that it was observed at the gene level also in Chardonnay by another group, by Laurent Deluc, higher levels of terpenes in this variety, and particularly at late stages at harvest. So this kind of is driving our attention. Now we are repeating similar studies with more applied treatments in Okanagan on Viognier grapes. And we have seen a similar effect on terpenes. However, like we have seen in this, sorry, I forgot this slide. This is a slide expressing like the gene level that, of the genes that control these compounds. And we actually see a modulation, upregulation of these genes, indicating that at the cellular level there is a higher expression of terpenes. It's not just a concentration effect. I was saying that we did not observe big difference in the wine quality, but the deficit wines were slightly more bitter, more color, and more retronasal intensity of the aroma. So more work has to be done before selling this as a good result or a bad result, the fact that terpenes can go up. And I'm concluding here. Sorry about being a bit late. So we have studied a lot in history the red grapes, and we know that flavonoids, anthocyanins can be directly modulated. We can increase the concentration of tannins, but not really the biosynthesis. We have seen, I think for the first time in Merlot, some increase of some volatiles. We don't know exactly how important they are for the quality of Merlot, although we see some positive effect on the aromatic profile of the wines. And finally, white grapes is an interesting topic, much less investigated. Different metabolites are important, and it's promising that terpenes might be modulated by deficit. However, we have seen also other phenolics like cinnamic acid, benzoic acid going up that could be bitter. So we don't know exactly, but I think we will know more in the future about how managing irrigation can affect the quality of white grapes. And I think I'm done. This is my lab and collaboration with Mark Matthews here about the Cabernet trial, and with the Institute of Edmund Mach that does metabolite profiling in north of Italy, some people in Israel doing profiling of primary metabolites that I didn't show, and my former university of Udine. So thank you very much for the attention. Yeah, sure.
So thank you, Simone. We have time for one or two questions. Yes?
Did you do sensory on the 2011 Merlot?
Sensory on?
Yeah, the sensory on the 2011 Merlot.
Yeah, so I didn't show, actually probably is, let's see if I put the slide here. No. Okay, so yeah, and 2011 was consistent with 2012 Merlot, and is consistent with what we find at the metabolite level. However, more severe is the stress. 2012 were a bit more severe. We see more effect than 2011 that was milder. On Tokai level, I showed 2012 data because 2011 were very different. It was a late deficit, 2 weeks before harvest. And actually it's what everybody was thinking was good because you get to concentrate some of your aromatic and so on. But we didn't see, in that case, in Tokai 2000 level, big difference when the deficit is severe and late during the season. We have seen big differences when the deficit was early and prolonged during the season. Oh, maybe that is last.
On the Sauvignon bird that you experiment with, do you see any effect on thiols precursors?
We haven't, yeah, so I understand what you mean. We haven't measured thiol precursors, yeah. Because thiols might be, it's not so easy to measure thiol precursors, so we haven't done that, but it's possible. And actually, I know that other people in Slovenia, that is the next door viticultural area, that my region is working on this Sauvignon Vert and thiol precursors, but I don't think they work on water depth, so I cannot answer this question.
Okay, thank you, Simone.
Yeah.